Hi. Last week in class we talked about passive transport. Passive transport is the movement of molecules across the cellular membrane from an area of high concentration to an area of low concentration. Today we're going to talk about active transport, which is sort of opposite of passive transport. Active transport is going to go from an area of low concentration to an area of high concentration. It's going to move against the concentration gradient, and it's going to require the cell to expend energy in the form of ATP. Active transport is going to move small molecules or ions across the membrane and is generally carried out by transport proteins or pumps that are found in the membrane itself. Larger molecules and clumps of materials can also be actively transported across the cell membrane by processes known as bulk transport. So we know that active transport requires the use of ATP. Okay? ATP is the form of energy that your cell uses. So our first example of active transport is an ion pump. This is when a transport protein moves an ion across the membrane against the concentration gradient using ATP energy. Our example that we're going to talk about is the sodium-potassium pump. And this pump plays a key role in electrical signals that move along your cells. So here's an example of the sodium-potassium pump. You can see the diagram there. Notice where there is more sodium and less sodium. Notice also where there is more potassium and less potassium. So where is there more sodium? Well, we see a lot of sodium here, and we see that it is on the outside of the cell. Where is there more potassium? We see that that is on the inside of the cell. So the next thing that we want to look at is which way these ions are moving. We see that the potassium is going from the outside of the cell to the inside of the cell. So it's moving from an area of low concentration to an area of high concentration. The sodium is moving to the outside of the cell, but it is also moving from low concentration to high concentration. So we say that it is moving against their gradient or up their gradient. And yes, this requires energy. The next type of active transport that we're going to talk about is endocytosis. This is an example of bulk transport. Endo means enter, and endocytosis is when the cell is moving large particles into the cell by the cell membrane engulfing the substance into a vesicle to bring it inside. So here we have the outside of the cell, and we have these molecules. The cell membrane wants these molecules, and so it will form a vesicle around these molecules, and then it will bring it into the cell. Then that vesicle can go to wherever it is needed in the cell. Exocytosis is another type of bulk transport. Exo means exit. Exocytosis is when vesicles carrying cell products fuse with the cell membrane, and those products are then released outside of the cell. So here we have our example. Here is a vesicle with material inside of it. The vesicle moves towards the membrane, and it fuses or connects with the membrane, and then it releases the products to the outside of the cell. So looking at endo and exocytosis, we want to know which type of cytosis would increase the size of the cell membrane. Hopefully you said exocytosis, because exocytosis, the vesicle already has a membrane around it, and then it goes to the plasma membrane and it connects with it and it makes it larger. In your review, in your packet, please take a few moments here and answer the questions. Okay, let's go over the answers. Osmosis is a type of passive transport that does not require energy. Endocytosis is active and it does require energy. Ion channels, passive, no energy. Facilitated diffusion, passive, no energy. Exocytosis, active, requires energy. And sodium-potassium pump is also active and requires energy. Now turn to page 29 and review active and passive transport. Stop the video and take a few minutes to answer the questions on the chart. Great. Let's check your answers. Okay, very good. All right, let's go back, sorry. Okay, now we're going to talk a little bit about cell size. And so what I first want to do is I want you to watch this 4-minute video about the biggest single-celled organism.
The elephant is a creature of epic proportions, and yet it owes its enormity to more than 1,000 trillion microscopic cells. And on the epically small end of things, there are likely millions of unicellular species. Yet there are very few we can see with the naked eye. Why is that? Why don't we get unicellular elephants, or blue whales, or brown bears? To find out, we have to peer into a cell's guts. This is where most of the cell's functions occur, enclosed by a cellular membrane that acts as the doorway into and out of the cell. Any resources the cell needs to consume, or waste products it needs to expel, first have to pass through this membrane. But there's a biological quirk in this setup. A cell's surface and volume increase at different rates. Cells come in many shapes, but imagining them as cubes will make the math easy to calculate. A cube has 6 faces. These represent the cell membrane and make up its surface area. A cube measuring 1 micrometer on each side, that's 1/1,000,000th of a meter, would have a total surface area of 6 square micrometers, and its volume would be 1 cubic micrometer. This would give us 6 units of surface area for every single unit of volume, a 6-to-1 ratio. But things change dramatically if we make the cube 10 times bigger, measuring 10 micrometers on each side. This cell would have a surface area of 600 square micrometers and a volume of 1,000 cubic micrometers, a ratio of only 0.6-to-1. That's less than 1 unit of surface area to service each unit of volume. As the cube grows, its volume increases much faster than its surface area. The interior would overtake the membrane, leaving too little surface area for things to quickly move in and out of the cell. A huge cell would back up with waste and eventually die and disintegrate. There's another plus to having multitudes of smaller cells, too. It's hardly a tragedy if one gets punctured, infected, or destroyed. Now, there are some exceptionally large cells that have adapted to cheat the system, like the body's longest cell, a neuron that stretches from the base of the spine to the foot. To compensate for its length, it's really thin, just a few micrometers in diameter. Another example can be found in your small intestine, where structures called villi fold up into little fingers. Each villus is made of cells with highly folded membranes that have tiny bumps called microvilli to increase their surface area. But what about single-celled organisms? Caulerpa taxifolia, a green algae that can reach 30 centimeters long, is believed to be the largest single-celled organism in the world, thanks to its unique biological hacks. Its surface area is enhanced with a frond-like structure. It uses photosynthesis to assemble its own food molecules, and it's cenocytic. That means it's a single cell with multiple nuclei, making it like a multicellular organism, but without the divisions between cells. Yet even the biggest unicellular organisms have limits, and none grows nearly as large as the elephant, whale, or bear. But within every big creature are trillions of minuscule cells, perfectly suited in all their tininess to keeping the Earth's giants lumbering along.
Okay, turn to page 19 and let's complete your notes on cell size. When a cell is small, it has a large surface area to volume ratio, while a large cell has a small surface area to volume ratio. So as the cell grows and its volume increases, that volume increases at a greater rate than its surface area. So its surface area to volume ratio decreases. Now, why is surface area to volume ratio important? It's important because it allows the cells to maintain homeostasis. And we know that cells need to maintain a stable internal environment so that they can survive. So which cells are more efficient? Well, hopefully you said the small cells, because that's correct. Remember that small cells have a large surface area to volume ratio, and that is best. There are some exceptions to the rule, and those are specialized cells that have had the ability and adapted their surface area to volume ratio by changing their shape. The first example is a long, thin shape. This is used by our nerve cells, or a neuron. Your other example is a cell that folds in the surface of your cell membrane. The example of the cells in your intestines, with the villi and the microvilli. Those little folds increase the surface area of those cells. And your third example is that plant cells are much larger than animal cells. They increase their surface area to volume ratio by having that large central vacuole which pushes all the other organelles to the edge of the cell, making those organelles closer to the cell membrane and thus efficient. So right now in your reading, or in your packet, I want you to turn to page 18, and there's a reading, Complete the Problem of Cell Size Reading. Read over it, highlight the important parts, and then answer the question. Once you've done your reading, your answer should be 6-1. If you have any questions, write them down, and when I get back to class tomorrow, I'll be happy to answer them. Have a great day.